All right, so the first question here was what, and as you see, the writing is difficult to read because of the bad photocopy, but it's definitely readable. What would most likely result if mitosis was not accompanied by cytoplasmic division? So, what part of the cell cycle is cytoplasmic division? That's really the question that you have to ask yourself. Cytoplasmic division happens during cytokinesis. We went through that, and we know that mitosis M phase happens, and then cytokinesis happens, and then you start again with G1, then S phase, then G2, that we've gone over many, many times. So what would happen if you went through mitosis, which is what? What is mitosis? The duplication of the nucleus. So at the end of mitosis, you have a single cell with two nuclei, as I've drawn numerous times, 46 each at the end of mitosis. So now let's go back and look at what the answer would be. What would most likely result if mitosis was not accompanied by cytokinesis or cytoplasmic division? Two cells? No, you're not dividing the cytoplasm. Not dividing. Let's circle this again, because that happens a lot. People don't see the knots, not accompanied by cytoplasmic division. So you're not going to have two cells. That automatically takes out A and B. So one cell. One cell with two identical nuclei, one cell without a nucleus. Well, why would there be no nucleus? The answer is C. And oddly, a number of people chose D, and a number of people chose either A or B. All right, let's move on. So at this point, you have to ask yourself, what was the problem with this question? Again, this particular question was written by the publisher. They used the word cytoplasmic division. You should know that what the cytoplasm is. You should know that that's cytoplasm. Everyone should know that. So I'm not sure why the dividing of the cytoplasm, why that would be any more difficult than saying cytokinesis. I guess if you're just memorizing words and not really understanding the concept, then it might be difficult to answer this question. But other than that, this question should be fairly straightforward. So here it says, and again, the photocopied went bad, and we're going to have to, I'll try to make sure that the photocopy comes out better on Monday, but it's still readable as I am reading it now. Which two processes are involved in mitotic cell division? It says mitotic. I think that's readable. Is it difficult to read? Sure, but it's certainly readable. So you know it's mitosis. You know it's not meiosis, since there's only two that we've studied, mitosis or meiosis. At this point, it's either one or the other, right? So what two processes are involved? So nuclear duplication, copying the nucleus, as we just said in the previous question, yes, check. Cytoplasmic division, check. Now, cytoplasmic duplication, making copies of everything in the cytoplasm, does that happen during mitosis? No, it does not. That happens in G1. That again is knowing your cell cycle. You need to know your cell cycle. If you didn't know that, if you haven't studied your cell cycle, you got problems. So you have to make sure you know what your cell cycle is. G1 is when you're duplicating everything, getting ready to start the mitosis or meiosis. S phase is when you're duplicating the DNA. G2 is you're checking everything and double checking it that you have enough of everything so you can split the cell. So this is not correct specifically because the cytoplasmic duplication happens in G1. Spermatogenesis, and did we even talk about this idea of spermatogenesis or oogenesis? Were those even words that we used in class? The answer is no. We did talk about the process, but not the details. So the spermatogenesis is the making of a sperm. Oogenesis is the making of an egg. They're slightly different, but they're just making a gamete. These, of course, would not be happening. There's two ways you can answer two ways you know that C and D are wrong. One, we didn't talk about it. Two, they are the product. This is the product. This happens after meiosis, not mitosis. Either way, C and D are wrong. A is the only possible answer. Here, which mitotic event will the chart in the chart occurs after the other three events have taken place? Well, here you either know the steps of mitosis or not. Again, mitotic cell division. Mitotic cell division. So the appearance of the spindle fibers, separation of chromatids by the action of spindle fibers. That would happen during anaphase. Disintegration of nuclear membrane and replication of the chromosomes. Now, what I said in class was that you can memorize the steps in order and know what each thing is happening. But if you understood why each thing happened the way it does, then you'd always know what the answers were. Now, of course, what do people do? They go home, they do the minimum, they memorize the definition of the words, but they don't really understand the process. So when they get a question like this, by the way, written by the state of Ohio, they can't answer the question. So let's, again, talk about the fact that as you have a cell, that's in the beginning of the first thing you have to do. If you have a cell that has chromatin before mitosis starts, all the DNA is unwound so it can do all the DNA replication and transcription translation. The first stage, the first step is you have to condense the DNA into chromosomes, and you have to dissolve, and they're two chromatids because they're copied, you have to dissolve the nuclear envelope so that you can separate the chromosomes. You can't separate the chromosomes unless you get rid of the barrier that holds them in the nucleus. And the centrioles have to move to the opposite sides of the cell if it's an animal cell, because animal cells need centrioles to attach the spindle fibers to. Then the spindle fibers form and attach to the chromosome, the sister chromatids, or specifically to the centromere, which is the center of the chromosome. So let's go through the answer choices. One of these happens last. The appearance of spindle fibers. Well, that probably happens. Oh, well, let's talk about the rest of mitosis really quickly. Then, of course, the next step is the chromosomes have to line up on the metaphase plate. Again, with the spindle fibers. You can look at the videos that we've done, hours of video of in-class instruction, examples, practice tests. Like, I don't know what, if you're confused, go to Khan Academy. I don't know what else to say. To me, this is just something, it's a matter of either you're putting in the effort or not, or And perhaps it's the words are confusing to you, but I just don't understand why they're confusing. So if there's something that is confusing, please feel free to Jupiter Grade message me a specific vocabulary word or concept but please don't ask me to go over mitosis again. We've done it 3 or 4 times. And again, Khan Academy goes over it. You have to put some effort into this, guys. You have to. So during metaphase, they line up, right? During prophase of mitosis, they condense and the spindle fibers attach. Metaphase, they line up in a line on a metaphase plate. Then in anaphase, the spindle fibers, they're attached to the centrioles in animal cells. They're attached to cell walls in plant cells, right? They separate this in anaphase. They separate the sister chromatids. Remember, they look. I keep telling you, they look like little shrunken heads getting pulled to opposite sides of the cell. So, and then of course, at some point, if you think about it, you have to have 2 nuclei, 46 chromosomes over here. The nuclear envelope reappears, and you're done with mitosis. And the next thing is, as we said earlier in the previous slide, we do have to do cytokinesis. You have to split the cytoplasm, because right now you have 2 nuclei in there. The next thing that happens is the pinching off of the cytoplasm with 46 chromosomes, exact copies of each in each cell. And you end up with 2 cells with 46 in each. But this is cytokinesis. Mitosis ended here, right after you copied both, and it started when you condensed it. So mitosis is metaphase. You might say prometaphase if you like. Metaphase, anaphase, and telophase. Those are your 4 steps to mitosis that you have to know. And again, if you know the process, you didn't even have to know names of the process. You just need to know the process. How do these chromosomes separate? So appearance of spindle fibers. Is that going to happen before or after you break apart the nuclear envelope? Well, you can't make these spindle fibers unless you get rid of the nuclear envelope. You have to get the nuclear membrane out of the way so that you can make the spindle fibers. So it can't be B. So either you memorize the steps and you know prophase is first. That means the disappearance of the nuclear envelope. Then the spindle fibers form, and the centrioles move to the opposite sides of the cell. Then the chromosomes line up in the middle. So separation of the chromatids. Separation of chromatids by action of spindle fibers. Well, that can't happen until after you have the spindle fibers. Spindle fibers can't happen until after you disintegrate the membrane. And the replication of the chromosome happens in S phase. So that was the first thing that happened. So the first thing that had to happen is this. It's not mitosis, right? This had to happen before before mitosis even began. Then you had to get rid of the nuclear envelope, so that's 2. Remember, this happened in S phase. And the disintegration, this happened in prophase. Then the appearance of spindle fibers had to happen before you could separate the chromatids. Now look at the logic here. More than one person chose the appearance of the spindle fibers as the answer to this question. This says which one happened after the other 3. So how are the spindle fibers going to appear before the spindle fibers separate the chromatids. It can't happen. How some people chose D. How can copying the chromosomes happen after you separated them? Like, you have to copy them before you separate them, don't you? You have to have two before you can separate the two. So so the third thing that had to happen had to be the appearance of the spindle fibers. And finally, the answer, the last thing that had to happen of the four things listed here, is the separation of the spindle fibers. So the answer is B, separation of the sister chromatids by the spindle fibers. All right. So the phases below describe several events that occurred during the process of mitosis. Attach a double-stranded chromosome. Again, is the photocopy horrible? Yes, but let's move on. Let's get over that. It's readable. How do I know? Because I'm reading it. I did not write it, and I didn't read it before I looked at it to identify the answers. So I had no advantage in reading this. Is it not easy to read? Is it perfectly clear? No, but it is. You can tell what the words mean. The phrases below describe several events that occurred during the process of mitosis. Attachment of double-stranded chromosomes to the spindle apparatus. Formation, and by stranded, this wording is really weird. The bookmaker used it, double-stranded chromosomes. You know, I don't like that term. I think what's obvious based on the answer choices they gave you is they mean the chromatids, two sister chromatids. They're trying to use words that, when they say double-stranded, they mean two sister chromatids. I don't like it. It's one of the reasons I gave. There's a couple questions that I felt were misleading in the way they were worded. This is one of them. That's why I gave everybody 20%. I gave everyone, 10%, 2 questions correct on the exam as part of your curve because of the odd wording. I didn't like it. I would not have chosen to use double-stranded myself, but that's the way they phrased it. Attachment of the double-stranded chromosome to the spindle fiber apparatus. And this is mitosis. Formation of single-stranded chromosome, which are moved to opposite ends. Again, I would not have used this term either. I don't like it. They mean chromatid. Formation of the chromatid, which are moved to opposite side of the cell. So this is obviously anaphase. This at the first one here, attaching the double stranded to the spindle fiber apparatus, here you're talking metaphase. Disintegration of the nuclear membrane, that's prophase. And nuclear membrane formation around each of the sister chromatids forming 2 new nuclei, that's telophase. And synthesis of spindle fiber apparatus. Now that had to happen before you attached the sister chromatids to it, and it had to happen after you disintegrated the nuclear envelope. So now again, what do they want you to do? They want you to put these steps in order. Well, the first thing that had to happen is you had to disintegrate the nuclear envelope. From these 4, this had to be number 1. So C had to be number 1. How could you do any of these other things if you don't get rid of the nuclear envelope? Well, right away, test-taking skills 101, the only one of these that has C as number 1 is this answer. So D is the answer. That fact makes A, B, and C wrong. The fact that you have to disintegrate the nuclear envelope. But if you want to know the order, 2, right? You make the spindle fibers. 3, you attach the double-stranded chromosomes. 4, you separate them, and 5, you make 2 new nuclei. If you want the order of the events, there they are. They are prophase, metaphase, anaphase, telophase, and then that's the end of mitosis, and you have cytokinesis. So what did some students do? A lot of students, actually. You memorize the words prophase, metaphase, anaphase, telophase, but you didn't know what the heck was going on in each of the steps. So that's your difficulty. That's the problem. All right, this one says, the cell in the diagram below illustrates the stage of mitotic cell division. Again, readable, though not great quality. Letter B indicates what? Somebody actually came up to me and says, I don't know what, they said to me, I don't know what this is. And I asked them, well, why? Why don't you know what this is? I don't understand. We've drawn it several times in class. You've had it on 2 practice tests. You have it in the copy in your study guide. You did it in your lab. So why is it that you can't identify what's happening here? They said, well, I didn't pay attention to the pictures. Wow. I don't understand. There's a reason we spent a day doing the lab. There's a reason we spent time drawing these on the board. And a reason why I begged you to make index cards. So, in any case, these are the spindle fibers. There they are. These are, I hope it's obvious, these are chromatids. The same person that asked that said, well, what are these black things? They're sister chromatids. They've been separated. So the separation of the sister chromatids are, and by the way, this young lady could recite the definitions of the words. So she knew the word definition, she knew the definition of chromatids, but just didn't know what it was. And I'm like, well, why did you think we spent so much time drawing these things and talking about them and me explaining the process to you? Because I knew what the state wants you to do. So spindle fibers, they separate the sister chromatids. In any case, at the very end, what are they going to attach to? I think the analogy I made is you're standing outside. If someone's car breaks down and we take a rope outside, what do we have to do? We have to attach the rope to 2 ends in order to pull the car. Well, these spindle fibers are the rope. They have to be attached at both ends. One end is to the sister chromatid. The other end has to be attached to the centriole. These are attached to the centriole, and that's what's pulling them over. So the things that are moved to each side during prophase, the things that move to each side of the cell, only in animal cells, because plant cells don't need centrioles. Why? What are they going to attach the spindle fibers to if they're separating their chromatids? I'm hoping you're saying cell wall. But animal cells don't have cell walls, so you need a centriole to do that. So the centrioles separate, and so that's your answers to centriole. Let's go through the other answers to make sure. Letter B is indicating the paired chromosomes. The paired chromosomes are here and here. They've been separated. The sister chromatids have been separated, and so that is not the answer. Endoplasmic reticulum. Okay. Actually, 4 people chose endoplasmic reticulum, and honestly, I just don't understand that. I don't know. Maybe that's just a panic answer, I guess. So it's not possible. Cell plate. The cell plate is what forms, and I drew this in class. Didn't call it a cell plate. That's why in the beginning of the test I told you they call it cell plate, and this is what the cell plate is. Plant cell has a cell wall, has a nucleus. If you made 2 nucleus at the end of mitosis, and you want to separate the cell, plants create a new cell wall before they separate their membranes. They create this cell wall. So they don't pinch off their membranes, they make a cell wall. This is called the cell plate. to separate these, to attach the spindle fibers before they do, this is obviously telophase, this is this is actually cytokinesis in plants, actually. But before they got here, we didn't have the nuclear envelope, so you see there's no nucleus here, it's just the cell. When they were separating the chromatids, the plants attached their spindle fibers to the cell wall on each side, and they use the cell wall to anchor their spindle fibers. I explained all this in class, but there it is again. So that's why this is not the answer. This does not have cell walls because it's an animal cell. If it has centrioles, which is what this is, then it does not have a cell wall because plants don't have centrioles. Okay, so the process of mitosis usually involves what? Well, let's read it. Chromosome duplication and synapsis. Synapsis is what's going on between the chromosome duplication. First of all, does not happen. You don't duplicate the chromosome during mitosis. That happens during S phase. So that part is wrong. Secondly, synapsis is what happens in meiosis. That's the creation. That's the crossing over business. So this is also during meiosis, not mitosis. DNA replication and separation of chromatids. DNA replication happens where? In S phase. Keep that in mind. So I didn't like this question at all, by the way. Tetrad formation and fertilization. Tetrads happen in meiosis. Synapsis is crossing over. Tetrad formation happens in meiosis. Both of these happen in meiosis. Reduction of chromosome number and formation of the cell plate. Well, usually is involved in mitosis. Reduction in chromosome number only happens in meiosis. So that one's wrong. Well, the only one that's possibly right is this one. But honestly, it's not right because they say DNA replication, and that does not happen during mitosis. So that's why I threw this question out. I didn't like the fact that they made this fundamental error and this again, this goes back to what I told you before. A lot of books, a lot of resources, they often put DNA replication in with mitosis, and that's just not correct. You do have to replicate the DNA. That does have to happen. You do have to make copies of the DNA, but that happens in S phase, not mitosis. So the diagram shown represent stages of cellular process. Which is the correct sequence of these stages? Again, pictures. A lot of people memorize the words. That's great, but pictures. The DNA is condensed. You can't tell what's going on here, but you know the DNA. You have chromosomes and you have a nucleus, and there's only one nucleus, so you know that this is prophase. So this has to be the first step, is prophase. Double checking. I mean, already I'm pretty sure I know the answer already because there's only one that has C as the first step. But let me double check, make sure they're not putting something else in there to trick me. Here, they're lining up in a line, so that has to be metaphase. Here, they're separating to each side of the cell, but they're actually all the way they're all the way on one side and on the other, and they're starting to form the nucleus again. That has to be telophase. That's the last step in mitosis. And then they're separating. There's no two nuclei yet. This has to be anaphase. So if you know what's going on and why, you got the answer right. If you memorized a bunch of words and their definition, then you got confused and you couldn't answer the question. So prophase is the first step. Metaphase is the second step. Anaphase is the third step, and telophase the fourth. Well, it's C, then B, then D, then A. The answer is C. All right, let's move on. What is the difference between the mitotic cell division in plants and mitotic cell division in animals? As we said, plants don't need centrioles. They have cell walls, and if you have a cell wall, you have to build the cell wall between the two cells before you can separate them at the end. But of course, that's cytokinesis. So let's see what answers, what questions. And I haven't even looked at these answers yet, so let's see. Well, I have, I guess, because I've chosen the answer, but I don't remember what the answers were. What is one difference between mitotic cell division in plants and mitotic cell division in animals? Chromosomes are replicated in plants, but not in animals. Well, first of all, chromosomes are replicated in both, right? And they're saying mitotic cell division, so they're not saying during mitosis, so they're not limiting you to a specific phase of the cell cycle. They're not saying only an M phase, right? So, but they're saying if you're doing mitosis, if you're doing cell replication to repair and to grow, then what's the difference between animals and plants? Chromosomes are replicated in plants, but not in animals? That's not true. They have to be replicated, otherwise it's not going to work, right? So this is mitosis, so you have to make copies, exact copies. The replicated chromosomes separate in plants, but not in animals. What? The sister chromatids don't separate in animals? That's not possible. See, there you have to know what? That replicated chromosomes are called what? Sister chromatids. Were those words used here? No. But you have to know what is meant by replicated chromosome. The cell plate divides cytoplasm in plants, but not in animals. Yeah. Cell plate, remember, and I said it at the beginning of the test, the cell plate is that in plants is what divides the cytokinesis in plants. So that is true. But double checking, making sure the last one's not somehow also correct. The nuclear membrane reforms in plants but not in animals. Of course not. Nuclear membrane reappears in both, as we've seen over and over again through all these questions. They're all the same. Which explains also why if you were getting one or two wrong, you were getting a lot of these wrong because you just don't understand the process. So this is A, and this is why I told you to come up and draw these. This was horrible. And I said, come up and make sure you understand what letters are where. Obviously, it's difficult to read that part, but everything else is readable. So it says, cell below is metaphase 1 of meiosis. There is no crossing over. So when this cell fully completes meiosis, now we're talking about meiosis, what gametes will be formed? Okay. So so see here, we're already separated. These are going to be pulled in which direction? Again, I drew these on the board. I drew this on the board, I want to say 3 or 4 times. You can go back and count how many times in the videos that I presented to you. You know the centrioles are on each side. You know you have spindle fibers that connect. This is meiosis, so that means that these homologs are going to be separated during this first step. So, okay. So if I'm going to separate the homologs, that means this is going to be out in one cell, in cell 1. This is going to be in cell 1, and this is going to be in cell 1. All of these are going to be in cell 1. So let's go ahead and draw that. Cell 1. It's going to have what? This with AA. There's going to be one with little b, and one with big D, big D. Then all of these are going to be in cell 2. So cell 2 is going to have what? It's going to have little a, and we did this in class. We have big B, and we have one that has little d. Okay. Well, what's the second step in meiosis? What's meiosis? Metaphase 2 of meiosis, anaphase 2 of meiosis. What are you separating? The sister chromatids equally to both cells. So let's go ahead and look and see what kind of cells you'd make. What kind of eggs or sperm, in this case blue for sperm, right? Let's see how many different kinds of sperms you'd make. Well, for the A chromosome, the chromosome with the A gene on it, you'd have one A here and one big A here. Why? How do I know that? There's only big A's. That's why they said no crossing over. Don't do crossing over so you don't get confused. Meiosis does do crossing over, but we're going to assume no crossing over happened here. So A and A, the D's, you got big A, big D. Or sorry, let's go with B's first. You have little b, one to each cell, and you have a big D, one to each cell. Well, already you know the answer because there's only one answer choice that has big A, little b, and big D. By the way, this is meiosis, and they say at the gametes. That means you have to have half the DNA. Notice there's 2 copies of each of these here, so that means this is wrong because haploid or gametes have half the DNA, so that's not right. This has double of everything as well, so again, that can't be right because by definition, gametes have half the DNA. So now if you didn't whittle this down to 2 possible choices right away, you don't understand what meiosis is doing. So that's a big problem for you. So you need to get on that. So then, if we take a look at what 2 eggs the female can make, with the A's, the only little A's can only give a little A. With the B's, you can only give big B. And the D's, you can only give little D's. I mean, look, this only has a big A to give. Only has a big A to give. Only has little D's to give. Only has big B's to give. So it has to be little A, big B, little d. So you see that the answer choice is D. If you knew what happens in meiosis, if you know what's happening in meiosis, then again, this is an easy question. So far there's only been one real problem, one question that had to be thrown out, and it was for everybody, and 2 questions that I felt were misleading, so I gave you everyone credit for those 2 questions. All right, let's go on. The diagram shown represents a microscopic structure observing to the process of cell division. They don't say meiosis or mitosis, right? What is letter B? Hard, again, difficult to read, but readable. That's B, and that's A. It's only A or B. So this is pointing to something in the center. Hopefully you know that's a centromere. How many times have we drawn this thing that looks like this? You've done a lab on it, activities, I don't even know how many different times over weeks of practice did we not say that this was a sister chromatid, this is a sister chromatid connected by a centromere. So they're pointing to this thing. What is this thing? Well, let's look at the choices. Centrosome? No. That's wrong. Spindle fiber? Well, spindle fibers in the cell. They connect to this, but that's not this. This is a chromosome. Chromatid. Well, that is the answer, but let's go on and check the last one. Cell plate? No, again, as I said at the beginning of the test, the cell plate's the wall that separates the 2 cells. The cell wall that separates the 2 cells, so that's not correct. So the only possible answer is sister chromatid, or chromatid. All right, that's number 11 on this version of the test. In the diagrams of mitotic cell division shown below, which is present in diagram B but not present in diagram A? All right, so let's go. So B is what? What is B? Looks interesting. This one's pinching off. A is pinching off. B is not pinching off. They both have nuclei. A has centrioles. B does not. B has this thing in the middle. square off as something in the middle. That's interesting. Centriole. Well, this is a centriole, so there's no centriole here. I don't see it. So this is in A, but not in B. It says in B, but not in A. So it's not this question, it's not A. Cell membrane. Well, if it doesn't have a cell membrane, it's not a cell. It's dead, right? So it's not a cell membrane. And if it doesn't have cytoplasm, so let's be clear. Right away, let me go back, and because this is important to notice this. Right away, cell membrane and cytoplasm, you knew that that was wrong. Both these cells have to have cytoplasm, have to have a cell membrane, or they don't exist. They're not cells. So it only gave you two possible answers. If you know what a centriole is, and I remember distinctly saying 4 or 5 times, you have to know the difference between centromere and centriole. It's going to be one of those things that are going to get you questions wrong on the test. I specifically said that repeatedly. And so you should know, and yes, it's not the greatest picture, but you should know that this is a centriole. Those are your centrioles on opposite sides of the cell to which spindle fibers were attached. So they are in A, but they are not in B. And cell plate, well, cell plate is this. That is the cell plate. That's what separates the two cells. And again, so let's think about it. What's the big difference? One has centrioles, one doesn't. One's square, the other one's roundish, one's pinching off, the other one isn't. You got a lot of clues here that tell you that this is a plant cell and this is an animal cell. All right. I told you A is true and B is false. I said write it in if you're confused about that. Right away, what are we looking at? We're looking at a karyotype. Let's see. You did an activity in class. You literally spend an entire period cutting these out and pasting them in. You counted the number of chromosomes in each spot. They looked very much like this. You counted each one. You did all 4 of these. You pretty much did during the activity. So when people tell me they don't know what they're looking at, I'm telling them not only did they not study, I'm wondering if you copied the work that you turned in to me, because everybody turned it in. Everybody got credit for it. So you must have copied this material or you forgot about it. I don't know what else to say. But we went over this. I told you it was going to be on here. We've said many times, every cell, this is the stuff we've said. This is the material that this is testing. Every human being has 46 chromosomes. They have 23 copies of each chromosome from mom and 23 copies from dad. So each chromosome should have 2. There should be 2 chromosomes. And they should have 2 sister chromatids at this point as well. You have to assume that those sister chromatids are there. And I told you that each one of these is a chromosome. I said that in class. I said during the test, I said each one of these is a chromosome. So and I also said in class, not during the test, but in class, I said repeatedly, when you have too many chromosomes or too few, most of the time you have a miscarriage. Sometimes when you have trisomy 21, chromosome number 21, sometimes you have 3 chromosome number 21s, and that leads to some kind of syndrome. In this case, it would be Down syndrome. But most of the time, if you have more than 2 chromosomes or only 1 chromosome, the fetus is miscarried. So, knowing all that, and by the way, there was literally a practice, every single one of these questions, there were practice test questions on the topic. And this particular one, someone asked me about it this morning before the first period test from another class. So they actually came up and asked me what was the answer to the question, and to explain it to them, and I did. So I don't know. I don't know what's going on here, but I just get this lack of something. Something's missing. We have to figure it out. So let's take a look at this. There are 46 chromosomes in this karyotype. I said each one of these is a chromosome. So if you just counted, it says this is a karyotype. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Now I cut some of these off, so let me look at the actual picture. So 3, 6, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 33, 36, 39, 42, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 60, 61, 62, 65, 66, 67, 68. So I counted 68 chromosomes. It's actually 69 chromosomes. I see there's a chromosome here. What is that? That's a Y. That's the X. Even if you found that hard to read as a Y, and honestly, if you really tried to look at it, you'd see it's a Y. I really don't see an excuse on that. But you know that there's an X and there's a Y. So there's two X's, and then there's a Y. All right. So those are your 23 chromosomes. There's 3 copies of most of these. There's 69 total. So those are the things that I looked at, just analyzing what I just saw, just reading it, counting. So now let's go ahead and take a look at this. There are 46 chromosomes in this karyotype. Are there? I just counted them. I literally counted 69. So that's not true. That would make this one false. So it's B. Male child. Well, has a Y chromosome, so it's male. True. So that's A. There are multiple trisomes in this individual. I told you in the beginning of the test, trisomies mean 3. 3. So there are many copies of 3. That's correct. That is true. True. A. The haploid number of this karyotype would be 34 or 35. Well, what's half of 29 or 69? It's either 34 or 35. So that's true. You can't have 34.5. You can't split these in half. This individual may have been miscarried. As I said earlier, most trisomies lead to miscarriage. I said that in class, so that is true. This one, obviously, this is the Griffith experiment. So the RNase was added to the test tube to make sure RNA was present. What? RNase gets rid of and this one I wrote because I felt I wanted to give you an easy test question. RNase gets rid of RNA. If you studied the Griffith experiment, paid attention to anything we did in class, did the case study that I sent home, looked at any one of the 4 videos from years past that have been posted, you know RNase gets rid of RNA. So that's not a possible answer. The heat killed, and by the way, this is clearly photocopied well. The heat killed was injected into the rats to verify that the heat killed bacteria did not kill the rat. That's true, but let's make sure the rest of them, that there's no other confusing point. The heat killed smooth was added to the rough to show that they would not kill the rats. No, the whole point was that you wanted to see if the heat killed cells would transform the the rough cells into smooth cells, and in fact they do. So that's not possibly an answer. So A is not an answer, and C is not an answer. Obviously, now the above is not an answer because we already said B is an answer. So the answer is B. If you're confused about that, please review your notes. So this question was easy, these two. So when you look at 19 and 20, if you got it wrong, you really don't understand semi-conservative replication. So when we're looking at this, we have two nuclei, and I told you they're dark. I even gave you a hint. I pretty much told you the answer. The two original DNA strands, I labeled them, and I continued to label them throughout. So these two old ones are together. That's called conservative. So that would be wrong. So let me do this in red. This is conservative replication. So instead of asking you about the radioactive nitrogen and the non-radioactive nitrogen, the heavy nitrogen, the light nitrogen, and give you bands and all this stuff that was in the experiment, I went easy on you and I asked you I went easy on you and I asked you for specifically just the main idea that this is conservative replication. This was disproved by the experiment. This one's dispersive. Why is this dispersive?
Because it's different chunks.
Because it's in chunks. That would be wrong. And let me go ahead and do this as in red because that is wrong, right? So now over here, you see there's one strand, and believe it or not, the photocopy looks better than what I have here. One strand is original, the other one is new. That's semi-conservative. So there's the pizza. Somebody has to go get it. Mr. Mendoza's room, may I help you? All right. Here, girls, go take my ID. They're downstairs. Go get them. They're at security. If they want a tip, they need me to sign for a tip, they have to come up. Oh, look at this. All right, semi-conservative. So that's semi-conservative, dispersive, and of course the answer would be semi-conservative C. And I even lined it up, and you can see that these two, one of the old strands stays, the others are all new. What's the only way? That's semi-conservative again. This is what you expect. That's why you have two bands, one in the middle and one was the heavy and one was in the middle, and that was semi-conservative. So that was that one. Those are those two. This one's obviously conservative, and this was dispersive. So this should have been easy. The answer here was obviously C, and this answer was C. So again, if you got this wrong, you have to ask yourself, did I go over the experiments? Do I know what's going on during the experiments? So again, it's the process. Memorizing the definitions would help, but they're not gonna help you if you don't understand how these changes occur and what they mean. It's the so what that makes a difference. All right, so you got a second chance on Monday. I'll post this at the end of the day. Good luck.